Hi guys, so in today's video I'm going to take you through absolutely everything that you need to know to smash those AQA A-level biology exams when it comes to photosynthesis. We'll talk about the light-dependent reaction, the light-independent reaction, and also limiting factors that can affect the rate that photosynthesis progresses. So don't forget to like, comment and subscribe, and let's get straight into the content. So let's cover all of A-level biology photosynthesis. Absolutely everything that you need to know. So we'll cover the light-dependent reaction first of all. Now, these are some of the key terms and definitions you need. So first of all, we have reduction, which is the gain of electrons or protons. Now think about oil rig. So reduction is the gain of electrons or protons or the loss of oxygen. Next of all, we have oxidation. Now oxidation is the loss of electrons or protons, so oil, as in oil rig. So oxidation is loss of electrons or protons, or it can be the gain of oxygen. Redox, next of all, that means reduction and oxidation happening together, because when one thing is reduced, another must be oxidized. Coenzymes work with enzymes. NADPH, remember the P for photosynthesis, that's a coenzyme that transfers hydrogen in photosynthesis. ATP is the universal energy currency of the cell, and it stands for adenosine, which is the A, tri for T, phosphate for P, ATP. Next of all, we have adenosine diphosphate. This time it has 2 phosphates, not 3. Photolysis is the splitting of water using light. Phosphorylation is adding phosphate, so phosphorylation, adding a phosphate. Photophosphorylation is adding phosphate using light. Photoionization is ionizing using light, so the energy from the light will cause that ionization, which is the release of an electron. Hydrolysis, split it into 2, hydro meaning water, lysis meaning to split. It's splitting with the addition of water. Decarboxylation means to remove CO2. Metabolism is all of the chemical reactions in a cell or organism. Condensation means forming a bond with the release of water. Think about the condensation on a car window screen. It's when you've got water droplets on that window. ATP synthase is an enzyme that makes ATP. Think about the term synthesis, to make, or synthesizer, to make something. A chloroplast contains chlorophyll, and it's the site of photosynthesis, and we're going to label that in a moment as well. So there's some of the key terms you need to know about to master photosynthesis, especially the light-dependent reaction. So here's a little summary of the light-dependent reaction. We have sunlight coming in, and that provides energy for carbon dioxide to be used to form oxygen and glucose. We also have the uptake of water through the roots, and that gives us carbon dioxide plus water goes to oxygen plus glucose. And you might remember from GCSE that you did that simple word equation and then learnt how to balance it 666. Now this little graphic on the right, I love this because you can see the light coming in and you can see the water and carbon dioxide coming in, and then you can see the oxygen and sugars leaving. But it actually gives you that little bit more A-level knowledge when we know that ATP, NADPH, they're used in this reaction to form that sugar. And I'm going to go all through the detail you need to know on this, guys. But this is a really nice overview, so I really recommend that you draw this. Now, there's 2 main stages to photosynthesis. There's the light-dependent reaction, which happens in the thylakoid membranes of the chloroplast, and the light-independent reaction, which happens in the stroma. Now, don't worry if this seems like a lot of new key terms to you and it seems very difficult. We're going to break it down step by step for you. So, this is the chloroplast. This is where photosynthesis happens. And you can see we've got the intermembrane space here between the 2 membranes. We have an outer membrane around the outside, and we have an inner membrane on the inside. I highly recommend that you draw this, guys, because you may be asked to label diagrams of this in the exam. You may be even asked to label diagrams of an electron micrograph of this in the exam too. Now we can see we've got the granum there, which is basically a stack of thylakoids. And the thylakoids are like little frisbees that make up the granum. And then between them, we have these little connections here. These are called the intergranal lamellae. And we have this liquid around it, this aqueous fluid. And aqueous just means it contains water. So think about Aqua or Aquaman, that hero who's the hero of the water. So the stroma is the fluid. And then we've got the thylakoids, like we mentioned, that are those little frisbees of membranes, and they contain that all-important chlorophyll. Now, to summarise, the light-dependent reaction occurs in the thylakoid membrane, so those green-looking frisbees, and the light-independent reaction occurs in the stroma, so that fluid within the chloroplast. So learn the structure of the chloroplast to master photosynthesis. So, this is an overview of the light-dependent reaction. And this is how I always remember it. So we have light hitting photosystem 2. That excites 2 electrons from that chlorophyll. So chlorophyll has magnesium at the core. Light hits it and 2 electrons, ping, go up to an electron acceptor. Now, from the electron acceptor, the electrons then pass down through electron carriers to photosystem 1. Now, this is going to lead to the generation of ATP. So we have ADP plus PI, which stands for inorganic phosphate, forming ATP. And that ATP is that all-important universal currency of energy. Then, light hits photosystem 1, and 2 electrons are again excited, and they move to the next electron acceptor. And we can have cyclic photophosphorylation here, where the electrons just keep moving round and round in this little three-way system here, and that's going to generate you ATP. But if we have non-cyclic, meaning it's not going round in a circle, like we can see here, it then goes down some more electron carriers here, and it forms NADPH. So draw this diagram, guys. This is so important to learn, and it's not going to make sense right now if this is your first time learning photosynthesis. But by the end of this video, it will make sense. So trust the process, guys, and learn it step by step with me. Now, in the background, we also have photolysis or photolysis, meaning to split water using light, because photo means light. So we have water, which is H2O, breaking down into 2 hydrogen ions, which means they've lost an electron to become positively charged. Now, those hydrogen ions, they're going to combine with the NADP, and I've highlighted that in red here. So you can see we've got NADP plus 2 electrons and 2 hydrogen ions, and that forms NADPH, which is going to be really important for the light-independent stage later on. Now, we can also see that water is broken down into 2 electrons, and you can see the electrons are there and there. So they're going to replenish photosystem 2 on the left. And we also have oxygen, which will be released from the stomata of the leaf. And if you're not familiar with the structure of the leaf, I highly recommend going back and revising that, guys, once you've watched this video, because learning about the structure of the leaf will really help you to understand photosynthesis. And that was in the Organisms Exchange Substances with Their Environment unit in first year A-level, or AS. Okay, so I've drawn an arrow here. We've got our 2 hydrogen ions from water combining with NADP, and we've got our 2 electrons combining with photosystem 2 to replenish those lost electrons. So let's go through it step by step, because like I said, guys, this is a difficult process. We've got to break it down. So number 1, light hits chlorophyll at the base of photosystem 2. And it's a bit weird that photosystem 2 comes before photosystem 1. And 2 electrons are then excited to a higher energy level. The electrons are accepted by an electron acceptor, and we can see that here. So that's our first phase there. Stage 2, the electrons pass down an electron transport chain, and the energy released drives the formation of ATP via photophosphorylation. And that basically just means adding a phosphate, phosphorylation, using light, photo. So we can see that there, we've got ATP. So we've added a phosphate to ADP to form ATP, and the T stands for tri because it's got 3 now. Now the protons move through something called ATP synthase, and that's going to drive the creation of ATP, catalyse the formation rather. Now number 3, light then hits photosystem 1 and electrons from chlorophyll are excited to a higher energy level and they're accepted by a second electron acceptor. Now number 4, the electrons may then either replenish the first electron acceptor, so they can go back to that one, and that will be cyclic photophosphorylation, so it will be going from PS1 to the electron acceptor to the next electron acceptor back to PS1, which is cyclic, or they can then carry on to that NADP, and then it will be non-cyclic because that breaks the cycle, okay, so it breaks the cycle, and that will then lead to the creation of NADPH, which is needed for light-independent photosynthesis, which is where glucose is made. Now, 5th, photolysis, that's where light splits a molecule of water to form 2 electrons, 2 protons, and 1 oxygen atom. And the protons combine with NADP and the electrons from PS1, so photosystem 1, to form NADPH. Now, number 6, the NADPH then enters the Calvin cycle in the stroma of the chloroplast, so that's the light-independent phase. And remember, when we were labelling the chloroplast, we said the light-dependent phase, so the one that needs light, happens in the thylakoids, and the light-independent phase, so the Calvin cycle, happens in the stroma. Again, don't worry if you haven't got it yet, guys. This takes time to build the steps you need to master this topic. Every single year, my students say that this is challenging. Now, here's a note for you as well. Cyclic photophosphorylation just involves photosystem 1 and it only produces ATP. Non-cyclic involves the full process. And some key things for you to know about the electron transport chain. That's going to crop up again when we learn about respiration. So make sure you understand that electrons passing down protein carriers are called the electron transport chain. Okay, so let's go into the electron transport chain in a bit more detail. Now, let me just flick back here and we can see number 2, the electrons pass down an electron transport chain. Well, that's here. That's where we've got ATP being synthesized. So that's where our electron transport chain is. So the energy released by electrons as they move down the electron transport chain transports protons into the thylakoids from the stroma. So we can see here we've got our electrons arriving here, so they're bringing with them some energy, and that energy is going to drive the flow of hydrogen ions into the thylakoid, so into those green frisbees from the stroma. It's going to drive the flow of protons, which are basically just hydrogen with an electron knocked off, into the thylakoids. Now, there will now be a higher concentration of protons in the thylakoids compared to the stroma. And you can see here I've represented that with loads of H+ ions in the area labelled the thylakoid, so within the green frisbees. And that's why it's so important to label the chloroplasts, so you can visually see where this process is happening. Now, in other words, we've got a concentration gradient of protons. So naturally, those protons are going to want to flow from a region of high concentration to low concentration. So our next step is that these protons will then move through a chemiosmotic gradient, which is basically where we've got chemicals and water. So chemi meaning chemical and osmotic meaning water, gradient through ATP synthase. And that's shown in orange here. So we've got this, it's basically like a carrier protein, but it's also an enzyme. So as those protons flow through it, it's going to cause ATP synthase to generate ATP from ADP and that inorganic phosphate. So ADP plus PI goes to ATP. So that's going to result in the formation of ATP, which we've got in our star here. So protons flow through, we get lots of ATP, which is that universal energy currency molecule. So to summarise then, the light-dependent reaction takes place in the thylakoid membranes, which is the grana and the intergranal lamellae. And remember, grana is the plural, granum is the singular. Photophosphorylation is where protons flow through ATP synthase at the end of the electron transport chain, forming ATP from ADP plus PI. Photolysis is where light energy is used to split water into 2 protons, 2 electrons, and half an oxygen. The end products of non-cyclic photophosphorylation are ATP, NADPH, and O2. The end products of cyclic photophosphorylation are just ATP. So well done, guys, you've just taken a step forward to mastering one of the trickiest topics in A-level biology. So we mentioned the light-independent reaction. Now let's dive into it. So I've got a really nice Calvin cycle here, and don't forget to like, comment and subscribe, guys. It means the world to me. So key definitions to go through next of all. Number 1, we've got ATP, which stands for adenosine triphosphate, and that's the molecule that provides energy. Or it can provide phosphate, because sticking a phosphate on a molecule can sometimes make it more reactive. RUBP is our next key term, which stands for ribulose bisphosphate, but don't worry, AQA do not need you to remember that for the exam. You can just write RUBP. Now, ribulose bisphosphate, the O's might have given it away, but it's basically a sugar. And it's actually a pentose sugar, meaning it's a 5-carbon sugar. Rubisco is the enzyme that catalyzes the carboxylation of rubisco. Now, I'm throwing a lot of technical terms at you here. What does carboxylation mean? Well, carboxy, it means carbon and oxygen. So it basically means adding carbon and oxygen to RUBP. Carboxylation, adding carbon dioxide. G3P next of all, that stands for glycerate-free phosphate, but G3P is fine for the exams. And that's basically a free carbon acid. We've got TP, which is triose phosphate, which is a free carbon sugar. NADP is a coenzyme that picks up a H plus ion, so a proton, from H2O in the light-dependent reaction. Next of all, we have NADPH, which is a coenzyme used to reduce G3P to TP, so glycerate-free phosphate to TP. And that's also known as reduced NADP. And earlier in the video, we spoke about what reduction was, and we remembered the mnemonic oil rig. So oxidation is the loss of protons or electrons, or the gain of oxygen, and reduction is the gain of protons or electrons, or the loss of oxygen. Now, the stroma is that aqueous matrix we labelled in the chloroplast earlier, and it's the location of the Calvin cycle. Redox reactions are where we have the reduction, gain of electrons or hydrogen ions, and oxidation, which is the loss of electrons or hydrogen ions. Light-dependent reactions happen in the thylakoids, and this produces NADPH and ATP. And finally, we have the light-independent reaction, which happens in the stroma, and you'll often see this written about as the Calvin cycle. And it basically is where the formation of organic substances such as glucose happens, and that uses carbon dioxide, the NADPH from the light-dependent reaction, and the ATP from the light-dependent reaction. So we can see we've got the light-dependent reaction, and that's firing in NADPH and ATP into the light-independent reaction, and then we have ADP and NADP without the H going back into the light-dependent reaction. So they rely on each other. And if you look at the image that I showed you on the first slide of this video, you'll see that it makes more sense now, guys. So as we go through, you'll be getting better and better at this. So we've got this beautiful Calvin cycle here, guys. And I always like to start my Calvin cycle with RUBP. And I want you to be able to draw this from memory, guys. Because if you can draw this from memory, you are a step closer to smashing those AQA A-level biology exams. So first of all, we have RUBP. It combines with carbon dioxide to form 2 times GP. So we've got a 5-carbon RUBP going to 2 lots of free carbon, so 6 carbons total. We then go from GP to TP, and that's going to form ADP. So we're basically going to have the breakdown of ATP, and it's also going to form NADP. Now from the TP, we then go back to RUBP, and that uses another molecule of ATP, which we get from the light-dependent reaction, and it forms organic substances such as glucose, but it could also form things like amino acids or glycerol. So we go from 5-carbon RUBP, 1 carbon CO2 comes in, 5 plus 1 equals 6. So we have 6 carbons at the GP stage. That goes back to 6 carbons, which is our TP, and then it goes back to 5 carbons with the RUBP. So we can see that we get 1 carbon locked into the plant per turn of the Calvin cycle. So it would take 6 rotations of the Calvin cycle to make 1 glucose, because remember, glucose is C6H12O6. So we have that 1 carbon going towards our sugars such as glucose. So let's break this process down, because to learn tricky processes like the light independent reaction, the light dependent reaction, it requires repetition and practice. So number 1, CO2 combines with RUBP in a process called carboxylation, adding carbon dioxide, because carbon dioxide is made up of carbon, carbo, and oxygen, oxy. Rubisco catalyzes this reaction. Rubisco will be there. Now number 2, 2 molecules of GP are formed, and we can see that here. So Rubisco catalyzes this reaction, and we can see that there with our 5 carbon RUBP combining with carbon dioxide in carboxylation to form a 6 carbon 2 times GP stage. 2 molecules of GP are then formed in stage 2, and we can see that happening here. Now next, NADPH is oxidized using 2 times GP to 2 times TP using energy from ATP hydrolysis. So that universal energy currency has been broken down, releasing a packet of energy that drives the oxidation of GP to TP. So we can see that happening here. 2 times GP goes to 2 times TP using ATP. Now number 4, some TP is used to form molecules like lipids, starch, cellulose, and amino acids, but the majority reform RUBP using energy from ATP, and that allows the cycle to keep going and going and going. But don't worry guys, thousands and thousands of these cycles are just going round and round and round constantly in every single chloroplast, so it stacks up. Even though just 1 carbon is fixed from the atmosphere, we are getting so many cycles going that we can make some massive polymers like starch. So we can see that here we have the organic substances such as glucose. So NADP and ADP then go back into the light dependent reaction to be converted back into NADPH and ATP. Now the light dependent reaction is all about producing NADPH and ATP so that we can make that all-important glucose. Now, Melvin Calvin, next of all, an absolute legend in the field of science. So Dr. Melvin Calvin was a Nobel laureate, which meant he won the Nobel Prize with his team, which was basically a £1 million prize and his name in the history books of science forever. And students like yourselves or myself when I was at school are going to be learning about him forever. Now, he discovered this cycle, and the way he did it was through Calvin's lollipop. And in AQA A-level biology, you can get asked all about this experiment, so it's important that we nail this. So let's look at it then. Melvin Calvin worked out the process of light-independent photosynthesis in his Lollipop experiment. And I've got a diagram of it here, so we can see, number 1, we had algae grown in a lollipop. So think about fish tanks when they get green stuff on the side of them. That is algae, which is basically single-celled plants that photosynthesize using chlorophyll. Number 2, radioactive hydrogen carbonate, and the isotope was C14, so we had a radioactive isotope, C14, that was injected into this lollipop, and the lollipop was like a big glass. It wasn't a sphere, it was like a flat circle, okay, filled with algae. In comes the radioactive carbon. Now number 3, algae uses the radioactive carbon dioxide to photosynthesize, and that's going to take in the carbon dioxide and that's going to carboxylate RUBP to 2 times GP. And remember, we've just done the Calvin cycle. Number 4, we had time intervals. So at time intervals, Melvin Calvin opened the tap at the bottom and released some algae into hot methanol. Now the reason there was hot methanol there was because it would instantly kill the algae and stop all chemical reactions. So no more light independent reaction, no more light dependent reaction. Now, next was the job of separating and identifying the radioactive compounds, as they will have been made using the radioactive hydrogen carbonate. And basically, what Dr. Kelvin did was use chromatography to separate out the different molecules to identify them. And as he separated it, this is what he found. So at 0, if he killed the algae, at 0 seconds, there wasn't enough time for the radioactive carbon to get integrated into the Kelvin cycle. So all that was radioactive in the methanol would be carbon dioxide. Now, after 5 seconds, there was radioactive G3P, which, remember, each molecule of G3P has 3 carbons, so it would have 3 of the carbon-14 carbons within it, and that would have been detected via chromatography. At 10 seconds, there will have been G3P and TP, which stands for triose phosphate. At 15 seconds, there will have been G3P, TP, and glucose. And at 20 seconds, there will have been radioactive G3P, TP, glucose, and RUBP. And this was how the cycle was built. So 0 seconds, we can see the CO2 there. 5 seconds, it goes to G3P. 10 seconds, it goes to TP as well. 15 seconds, it would go back to glucose. And 20 seconds, it would go back to RUBP. And that was how Melvin Kelvin discovered the Kelvin cycle operating in the stroma of the chloroplasts. So let's summarize this then, guys. The light-independent reaction uses NADPH, which is reduced NADP, and ATP from the light-dependent reaction to form sugar. And I always remember with NADPH, think about the P being for photosynthesis, because there's NADH in respiration, and it can get confusing. So NADPH is the one in photosynthesis. Next of all, RUBP reacts with carbon dioxide to form GP. Rubisco catalyzes this carboxylation. ATP and NADPH reduce GP to TP. Some TP regenerates RUBP. Some TP is used to make organic substances such as cellulose, starch, glucose, amino acids, nucleotides, and lipids. So this is how the plant makes important biological molecules for itself. And that is the light-independent reaction, guys. So now we're going to jump into limiting factors. And I'm going to show you this lovely diagram again now that I showed you on the first slide of the video. And hopefully it makes a lot more sense to you, because we can see we've got ATP going from the light-dependent reaction into the Kelvin cycle. We've got NADPH going from the light-independent reaction into the Kelvin cycle. And we've got the NADP and the ADP and PI going back into the light-dependent reactions. So we can see we've got the light-dependent reaction feeding into the light-independent reaction and vice versa. So they need each other. Now, I know you guys like all of the revision you need for a topic in one video, so that's what I'm providing here today. So we're going to talk about limiting factors next of all, which is basically the factors that can limit the rate of photosynthesis. So let's dive in. Photosynthesis is limited by 3 key factors. Number 1, temperature. Number 2, light. And number 3, carbon dioxide. And I'm sure you can already think about the temperature will limit the rate of enzymes. And we've just talked about Rubisco in the light-independent reaction. Light is obviously going to limit the rate that the light-dependent reaction can occur, because we need the light to excite those electrons from the photosystems. And then we've got carbon dioxide. Remember, that's needed for carboxylation of RUBP. So the process of photosynthesis will be limited by whichever of these 3 variables is the least optimal. So even if the other 2 are in plentiful supply, there will be one factor limiting the rate at which photosynthesis can take place. So for example, when carbon dioxide is the limiting factor, an increase in carbon dioxide concentration will therefore increase the rate of photosynthesis. As photosynthesis increases in rate, so will the rate of oxygen production and carbon dioxide absorption. And we can measure these 2 variables, so oxygen production and carbon dioxide absorption, to determine the rate of photosynthesis. And you know how much AQA love a practical question in the exams. So let me help you with that, guys. When the amount of oxygen produced and carbon dioxide absorbed during photosynthesis is matched by the amount of carbon dioxide produced and oxygen absorbed by respiration in the plant, this is known as the compensation point. Now, don't fall into the trap of thinking that plants don't respire. Plants absolutely respire too. All living things, apart from viruses, which are debatable whether they're alive or not, respire. So plants are no different. So if the rate of their photosynthesis matches the rate of their respiration, that is known as the compensation point. Now, at the compensation point, there will be no net gain or loss of gases. Normally, carbon dioxide concentration is the limiting factor in plants. Carbon dioxide makes up around 0.04% of atmospheric gas, and that's along with around 78% nitrogen and 21% oxygen, along with argon and other trace gases. Studies have found that higher concentrations of carbon dioxide increase crop yields by increasing the rate of photosynthesis. And interestingly, NASA also found that increased carbon dioxide reduces water loss by crop plants through transpiration. More carbon dioxide means more substrate for rubisco to combine with RUBP in the Calvin cycle. And if CO2 and light are in plentiful supply, an increase in temperature will lead to a direct increase in the rate of photosynthesis. So between 0 and 25 degrees, a 10 degrees Celsius increase will actually double the rate of photosynthesis. But again, we've got to think about temperature and the rate of enzyme catalyzed reactions. And the enzyme in question here is rubisco in the light independent reaction. So here's 3 graphs that you guys need to get used to. So we can see that as the light intensity increases, the rate of photosynthesis increases and then plateaus. As the carbon dioxide concentration increases, so does the rate of photosynthesis until it reaches its optimal rate and then it plateaus. And finally, with temperature, we can see that as the temperature increases, so does the rate of photosynthesis until it reaches its optimum rate. But then after this, the enzymes will denature and we have a sharp drop in rate of reaction. So you need to be able to, 1, describe these graphs, and 2, explain these graphs. This is another graph that you need to be able to describe and explain. And we can see here that we've got experiment 1, where we had 25 degrees Celsius and 0.1% carbon dioxide. That was at a faster rate. Experiment 2, we had 25 degrees Celsius and 0.04% carbon dioxide at a slightly slower rate. And experiment 3 at 15 degrees C and 0.04% carbon dioxide, and that was the slowest rate. Now, you need to be able to link the 3 experiments to your knowledge of the light-dependent and light-independent reaction. It's very possible AQA might ask you a practical question on photosynthesis, and it may include some data that looks like this in the exam. So, how could we find out the rate of photosynthesis? What is limiting the rate of photosynthesis for the beginning of experiment 1? And if there are low levels of sunlight, e.g. in winter, why would it not be cost effective for a farmer to heat a greenhouse? So how could we find out the rate of photosynthesis? Well, we could measure the volume of carbon dioxide absorbed or the volume of oxygen produced. And this is some gold for your exam technique, guys, because this is a classic way that you could get asked about photosynthesis in the exam. What is limiting the rate of photosynthesis at the beginning for experiment 1? Well, here, light intensity as an increase in light leads to an increase in photosynthesis, even though the temperature and carbon dioxide levels are controlled. So, if there are low levels of sunlight, e.g. in winter, why would it not be cost effective for a farmer to heat a greenhouse? So that's because if there's not a lot of light in winter, that would limit the light-independent reaction, because don't forget the light-independent reaction relies on ATP and NADPH from the light-dependent reaction. So we could increase the temperature, but the Calvin cycle would still be slow because it needs NADPH and it needs ATP. So to summarise next of all, guys, photosynthesis is limited by light, carbon dioxide concentration and temperature. Higher temperatures give Rubisco more kinetic energy and therefore a higher chance of a successful collision between enzyme and substrate. So Rubisco plus carbon dioxide, aka carboxylation, will only occur at high enough temperatures, and the key term here is enzyme-substrate complex. Next, increased light intensity will lead to greater levels of photoionization, therefore more photophosphorylation, which is the production of ATP, and more reduction of NADP for the Calvin cycle. Now I've hit you with a lot of words there, but basically photoionization means making ions using light, so when we have electrons moving, and photophosphorylation means adding a phosphate to ADP to form ATP. So if we have more production of ATP and more reduction of NADP to NADPH, that means the Calvin cycle can occur at a faster rate and therefore will have an increased rate of photosynthesis. So to sum it up there, basically, if we have more light-dependent photosynthesis, we can have more light-independent, which is where glucose is made. Higher carbon dioxide concentrations lead to more substrate for Rubisco, therefore more enzyme-substrate complexes and a faster rate of photosynthesis. Photosynthesis is limited by the factor that is least favourable at any given time out of light, carbon dioxide and temperature. And that's all we've got time for, guys. We've just done everything you need to know in photosynthesis, so I hope that was useful. Don't forget to like, comment and subscribe, guys, and I will see you in the next one.